Medicine Without Cruelty presents Understanding Human Biology Part 2 Tissues and Organs. Before we begin, as always, I want to start off with this quote. The greatness of a nation and its moral progress can be judged by the way its animals are treated. We are all working collectively to educate ourselves and move away from the atrocities of animal testing and move towards cutting edge human relevant techniques. So we learned in the last video that humans are made up of cells, tissues and organs, and the complexity of these structures can vary significantly across species. Animals don't replicate human biology, and that's a massive problem. We learned about cell organelles, so the substructures within a basic cell, which is the basic building block of all living beings. And we learned about three important structures: mitochondria, nucleus, and cell membrane. The mitochondria generates energy necessary for cells to function, playing a key role in metabolism and therefore drug metabolism. The nucleus contains the cell's genetic material and is responsible for regulating gene expression and cell division. Now, we're going to be revisiting some of these points later on in the series, so don't worry, we will be revisiting all of this knowledge again. And finally, the cell membrane controls what enters and what exits the cell, playing a critical role in drug absorption, distribution, and cell signaling. So just to remind you, they are the powerhouse, control center, and gatekeeper of the cell. So if we move forward, let's talk about tissue. Now, tissues are groups of similar cells that work together to perform a specific function. So we were talking about, we likened cells to Lego bricks in my last series. If we think about putting similar colored Lego bricks together, we can think of that as what a tissue is. It is a group of similar cells that work together to perform a specific function. So, for example, muscle tissue, they are made up of muscle cells that work together to enable movement. There are actually four main types of tissue within the human body. There is epithelial tissue. They cover the surfaces and lines all your organs, and such as your skin. And even the lining of your digestive tract is covered with epithelial, what's called epithelial. We have connective tissue, so it provides structure and support like bone and cartilage. We've got muscle tissue. We have nervous tissue. It transmits signals throughout the body and it's found within the brain and the nerves. Now, organs, they are the next level up. They are structures that are made up of multiple types of tissues that work together to perform complex, very specific functions. For example, the stomach, that picture in the bottom left of your screen, is an organ involved in digestion with muscle tissue to churn your food, epithelial tissue to protect the lining of your stomach, and connective tissue to support its structure. So all three different types of tissue are used together to create your stomach. Another example of tissues working together in an organ is your heart. So your muscle tissue within your heart enables the pumping of blood throughout your body. Connective tissue supports the structure, and nervous tissue helps regulate your heartbeat. These tissues collaborate to ensure the heart can pump blood effectively. So how are they all related? Now, tissues and organs are connected to each other in organ systems. Organs don't work in isolation. They are connected to other organs to form an organ system. For example, your digestive tract or your respiratory system, how you breathe and how you process your food. So if you think about your digestive system, your stomach, your intestines, your liver, pancreas, all work together as part of the digestive system to process food, absorb nutrients, and eliminate waste. And actually, there's communication between tissues and organs because we have nervous signals, hormones, blood flow, allowing them to coordinate activities. A simple example is when you're hungry, your brain signals to your stomach to prepare for digestion. Understanding how cells form tissues and organs allows scientists to replicate these structures in a lab, like organoids or organ-on-a-chip technologies. These models can mimic human organ function, enabling researchers to study disease or test drugs in systems which resemble actual human organs. Because remember, these therapies, these treatments we're designing are for humans. And so we need to make sure that we can resemble actual human organs. And so, just kind of a final point, that similar cells work together to develop tissues, and organs are made up of different types of tissues all working in unison. So, how do they help us survive? Cells, like I said, they are the smallest functional units of life, carrying out basic processes that keep us alive, like energy production, waste removal. Cells group together to form tissues. They're designed to provide shape, structure, and protection. And that contributes to the body's function. And finally, your organs made up of multiple types of tissues that allow body systems to perform specific functions, such as your heart, for example, or your stomach. Each organ has that specialized role which contributes to larger organ systems, like I mentioned earlier, your digestive tract, your respiratory system, so the system where you breathe, and your circulatory system, how blood gets pumped throughout your body. So, organoids are these 3D clusters of cells, and they are usually grown from stem cells. Before I begin explaining organoids, I need to explain stem cells. Now, stem cells are special type of cells with their unique ability to develop into many different cell types. So they're often called specialized or undifferentiated cells because, like any other cell, they haven't committed to a specific role or a function. But these stem cells can divide into muscle cells, blood cells, any kind of cell you want. And we usually have them, they're usually found at an early stage when we're embryos, because these stem cells turn into any cell type in the body. And we also have adult stem cells. So they're found in specific tissues like bone marrow, in the brain. And these cells are more limited, but they can repair and regenerate tissue in their specific areas. So organoids are clusters of these 3D clusters of stem cells that are grown into the organ of choice. So, say if we wanted to create a liver organoid, we would promote the growth, the differentiation, I should call it, of these stem cells into liver cells. These organoids can then replicate the architecture and function of real human organs. So, like I said, once we've grown the cells needed to form tissues, these tissues can then work together as an organ. Now, it's really important because scientists can use organoids to model specific human organs, such as the brain, liver, lungs. And then researchers can study how diseases affect organs or how drugs interact with specific tissues, providing an accurate understanding of human biology than animals could ever do. Just like tissues in the human body, organoids are made up of specialized cells that come together to mimic real organ structures. So, for example, a liver organoid contains liver cells that function similarly to how they would in the actual human liver, allowing scientists to test drug metabolism directly in human-like tissue. The actual organ on a chip is a microfluidic device, and microfluidic devices, you might have come across them already. They are at-home pregnancy tests or COVID-19 tests that you take at home. They are both examples of microfluidic devices. Basically refers to the manipulation of very small amounts of fluids, often nanoliters or microliters, through channels to perform very specific biological or chemical reactions. So in this case, we have a microfluidic device lined with living human cells, which simulates the environment of entire organs. So these chips not only recreate the cellular organization, but also the mechanical and the biochemical processes that occur in the body, such as blood flow or nutrient exchange. And it's really important because, unlike simple cell cultures or even animals, organ on a chips provide a dynamic environment where scientists can observe how cells behave under real physiological conditions. This is crucial for studying how diseases progress, how drugs are absorbed, or how toxins impact organs in a way that reflects true human biology. Now, while organoids recreate the structure of organs in 3D form, organ on a chip technology goes a step further by being able to mimic the functionality of organs, allowing cells and tissues to interact in a way that mimics how organs work in a human body. For example, a lung on a chip can replicate that mechanical motion of breathing. The liver on a chip model uses human liver cells, providing a relevant platform for testing drug safety in humans. So, like I said, how does it work? So, this chip is engineered with small channels to allow fluids like blood or drugs to flow over the cells, simulating that body circulation. That flow replicates how the liver cells interact with drugs and toxins in a dynamic environment. And then we can extract real time data, and scientists can monitor the liver cell response to drugs in real time, allowing them to detect any signs of liver toxicity early. And we actually, it enables accurate predictions of how the human liver would metabolize and respond to specific medications. There was a success story which showed how liver on a chip was able to predict drug induced liver toxicity where animals have failed. For example, drugs that showed no signs of toxicity in animals but were correctly identified as being harmful, that was identified on the liver on a chip model. So this has profound implications for pharmaceutical development as it provides a safe, reliable way to test new drugs before they enter human trials. Now, the development of liver on a chip technology is part of a larger movement to human relevant research methods that completely obliterate the even use of animal testing. We don't need it. By using human cells and replicating human organ functions, the liver on a chip models bridge the gap between lab-based research and real-world human biology, paving the way for effective and ethical drug development. And so, to sum it up, understanding human cell biology and physiology is the key to effective human relevant science. Animal suffering is completely preventable with modern science. We have human relevant models to protect humans from unsafe drugs and provide accurate results. And these methods eliminate outdated and ineffective animal testing. Thank you very much. Please like and subscribe, and looking forward to the next one.